This is our website, Biology 9 Chicks. To start learning about our experiment, press the button Start Learning or go to the pages here. If you press the Start Learning button, it'll take you to the introduction, which tells you about the Biology 9 we were learning, and the chick embryo shell culture vessel project that we are doing, which the introduction will explain what it is and what we hope to achieve by doing this project. To learn from the beginning, it'll take you to the DNA page and all about DNA, such as cloning, to genes, to gene traits and variations, to gene mutations. Once you're at this last page and there's no more buttons, you'll see that there isn't. You'll have to go to pages to check out another source of information, such as the cell cycle. The cell cycle talks about the stages of the cell cycle and what they do, such as interphase, mitosis, and cytokinesis. In here, you'll learn about how a cell is replicated and duplicated and separated. The cell and its organelles. The cell and its organelles explains what an animal cell is and what a plant cell is and the parts of it. And it includes a video that explains it in a more basic way for anybody just learning it out. Once you're there, you can go back to pages to learn about sexual reproduction and meiosis, which are relative to both each other. If you click on the sexual reproduction, you can go to meiosis and click onto that to see the meiosis page. Or you can go learn about asexual reproduction, which is done by bacteria and only requires one parent, unlike like sexual reproduction, and you can go vice versa to learn about sexual reproduction again. Once you're done this, you can go how a chick embryo is formed and see the procedures on how it all happens, such as how it goes through the mother hen and mitosis occurs to create an embryo. Here it explains the body at which the shell goes through, the embryo goes through, and the shell is added. These are parts of the shell and the membrane. The development here shows how an embryo will develop throughout the stages of 21 days in a more detailed way. If you want to look at just the milestones, you can always click down or just keep going. What a chick needs to survive is next by clicking this button, and it'll explain factors that you need to keep a chick healthy and have the best results for hatching. These reasons for death or poor embryos are all part of the incubation process. How to incubate eggs properly is here so that you can always incubate eggs at the right temperatures and the right way. This will show you what to do and how to do it. How to make an egg incubator if you don't have one or cannot buy one. Watch this video like we did to learn how to make an incubator. We had to use this video to make our incubator for our project. If you want to buy an incubator, here's some good quality brands that you can use. By pressing this button, you can go to factors that could affect the embryo, which shows factors that could affect the embryo as a whole. If you go to pages, it'll take you to purpose of the egg, which shows you the purpose of the egg and the parts of it and what it's used for, such as protection, nutrients, and oxygen carbon dioxide exchange, which is oxygen aeration. If you would like to keep going, you can see how to raise chicks after hatching in either a brooder or a shelter, a shelter such as a chicken coop, after a few weeks of them living, and what to feed them to keep them thriving. Once you've done this, and you learned the basis of what our experiment is based off of, you can go to the experiment lab report. In this shell cultured embryo page, you can see what it is a shell cultured embryo. These Japanese students, which we based our procedure off of, created a cup using a plastic film to hold the embryo. We did our procedure based off theirs, and if you would like to see their procedure and lab report, click this link to do so. If you would like to continue with ours, press the abstract to see a summarized version of what we did in the few weeks that we learned about chicks and the shell egg culture. The background now will show you all about we'll show you all about the past presentations used by other people and how successful they were and how unsuccessful they are, such as surrogate eggs and the cup method as well. Our hypothesis, which is if we take an embryo out of its natural shell and place it in a culture vessel, then it will grow and be beneficial to understanding the development of an embryo, is what we believe will happen. Materials and equipment used and solutions were all put here, and also a map of where we got our eggs from. To click onto procedure, it'll show you a procedure of pre-incubation, or how we made our homemade incubator for the embryos, and our cultured vessels, and the 3 different cups designs we used for each embryo. This video will show you a process of all the pictures we took. We also added oxygen aeration, so if the embryos did survive till day 17, we're going to talk about the percentage of pure oxygen and regular air we would give them, and all the experiments we might have tried if they did survive until then. Data results and observations were all put here in quantitative and qualitative ways, such as pre-incubation results day 1 to 3 and day 4 to 9, using pictures and writing our descriptions of what we see. These videos will show you development that we pointed out and looked at. By here you can see the end of our project and what the embryo became when it died. Analysts with things we thought that we had problems with, such as lack of observation, temperature, and sterilization. These were problems we thought that would have factored against the death of the embryo. Our conclusions stated what we did for our project and then how it ended up and the problems we had within it. We also did accept our hypothesis because, like we said, it was a beneficial experience to study the development of the embryo, and we do believe you can use this method to study other species such as birds or maybe reptiles, but the cultured vessel is not like a good substitute for hatching chicks in an agricultural use like we thought from the introduction. So if you want to go back to the introduction to see what we thought, but then saw how it ended up in the conclusion, you can always do that. Once you're done with learning and about our project and all the biology, you can go to resources to see where we got all this information from and where we learned the basics of our experiment from. If you want to have some fun, you can go to the quizzes and try what kind of chicken are you and have you been paying attention, which is a quiz on all the biology we learned. Thank you for listening. Bye.